All right, let's go ahead and get started. So last class, we focused primarily on innate immunity. Today, we're going to pivot and talk about adaptive immunology. I kind of gave the brief, bit reductive description of innate immunology as being fast but dumb and adaptive immunity being slow but smart. That dynamic of it being slow but smart entails a little bit more complexity within adaptive immunology. And so I'm going to try and make sure that we're having opportunities to try and stop and have a lot of opportunities for clarification. And again, if something doesn't make sense, that's not on you, that's on me. And so please ask any questions for clarification. And if you have a burning question, it's likely that someone else in the room might have the same question and would benefit from you asking it. So please, if something's not clear, or if even I haven't properly defined a blue term, please take the opportunity to try and raise your hand and ask for that clarification. So we're going to be talking about adaptive immunology against the backdrop of Guillain-Barré syndrome. A lot of work in immunology in general first has its start by trying to look at and explore really unique organisms that allow us to make really important discoveries on the basic science level in immunology, and that was particularly the case with the adaptive immunity. And they leveraged a really interesting organism called the hagfish. It's a jawless fish that actually only has an adaptive immune system. It lacks its innate immune system. And so it was an organism that was able to be leveraged to identify really fundamental pieces of adaptive immunity. And what's also interesting is that they produce these slime glands that are part of their first line of defense within their immune system. We have various layers of physical lines of defense that coordinate with our immune system: skin, mucus. And so for the hagfish, unfortunately for them, they have these slime glands that produce a really interesting slime. So for Guillain-Barré syndrome, it's an acute autoimmune polyneuropathy. So we talked about how our immune systems, their main goal is to identify self as self and foreign as foreign, to distinguish self versus foreign. An autoimmune disease, typically self is identified as foreign and is typically oftentimes, depending on where the autoimmune disease is located, or not where it's located, but what part of our body is identified as foreign, can manifest in different ways. And so a polyneuropathy typically means that there are neurological dysfunctions, multiple neurological dysfunctions are associated with this disease. And so in this case, there's a part of our peripheral nervous system that's identified as foreign here. And this is oftentimes the most common form of flaccid paralysis out there. The range of symptoms can vary, and it ranges from either innocuous to more severe. It can present as pins and needles and some nerve pain. It can manifest all the way up to severe muscle pain across different portions of the body. Can affect the ability to walk. Can affect eye movement and facial features. It can eventually affect really key autonomic functions, heart rate, blood pressure, body temp, and can eventually affect difficulty breathing. There's actually a few number of recent high-profile Guillain-Barré or GBS cases, including, I believe, the Texas Governor Abbott as well. And so while there's varying ranges of prevalence across countries, it's still something that can manifest in the U.S. So for Guillain-Barré syndrome, there's oftentimes, it is preceded by a specific infection. We'll talk about what that pathogen is. After the infection, there's usually a progressive phase where the symptoms will typically escalate over the course of several weeks. After that, it'll plateau and it'll typically reach resolution, but how long it takes for it to reach resolution can vary on the order of months to years. And in some cases, some patients can still have weakness on the order of several years after infection. Here, there's oftentimes there is a differential diagnosis that is associated with GBS. What that means is that there are the symptoms that are associated for differential diagnosis means that the symptoms associated with this disease can sometimes resemble or be similar to symptoms for other diseases. And so with here typically, and it's some of the same ones that some of them are some that we've already talked about. For example, botulism. It can also resemble heavy metal intoxication like lead poisoning, myasthenia gravis, polio, as well as tick bite paralysis. The etiology or the cause of GBS is typically caused by a specific bacterial infection, oftentimes by Campylobacter jejuni or C. jejuni. What ends up happening is that the pathogenesis for this disease is that it develops where our Schwann cells, these are the cells that wrap and encase around our peripheral nerves that produce the myelin sheath that help promote saltatory conduction. Those Schwann cells become the self that's identified as foreign and they're attacked. And so what ends up happening is that with the loss of these Schwann cells, there's a loss of the myelination around these peripheral nerves. And that is what ends up leading to the the effect on motor neuron function and motor nerve conduction. There's going to be some key terms that are going to be really helpful to define and describe before we really jump headfirst into autoimmunity, on adaptive immunity, forgive me. And so again, as I described earlier, autoimmune disease, and it always bears help, I think, repeating, the function of the immune system is to distinguish between self and foreign. It's also meant to also distinguish between, for what's identified as foreign, what's innocuous and what's dangerous. And so primarily autoimmune disease is where the self is identified as foreign and treated as an invading foreign pathogen that needs to be attacked. A key term whenever we talk about immunity is antigen. Antigen is typically any substance that's able to produce an immune response. And so in cases of autoimmune disease, the piece of our body or the whatever the molecule is or whatever the cell is that elicits this autoimmune response is referred to as the autoantigen. And so, for example, in GBS and Guillain-Barré syndrome, we talk about the Schwann cells as being part of the autoantigen there. In diabetes, the pancreatic beta cells are the autoantigen there. Type 1 diabetes is the autoantigen there. And what's important to note is that the antigen is typically like the entire pathogen. Usually, there's a specific feature on that antigen that is oftentimes the immune response triggering agent that's identified. That's typically referred to as the epitope. So, for example, if my left hand is an antigen, it's a substance that can produce an immune response. Perhaps the first carpal on my index finger would be an epitope. Any small feature on an antigen that can produce an immune response is typically referred to as the epitope. So when we talk about recognition of an antigen, there's going to be specific features or parts of the antigen that's recognized. That feature, that part that's recognized, is called the epitope. And we're going to talk about a few. We're going to talk about a few proteins that recognize epitopes, and it's going to make a little bit more sense. And we can try to apply it to a couple different circumstances. And so, for one example, a lot of people have gotten different vaccines over the years. There is, for example, a great example is that everyone can appreciate is the coronavirus vaccine. The coronavirus is this is a specific virus that has usually littered with proteins on its surface. For the coronavirus, the specific epitope that's oftentimes recognized as antigenic is spike protein. So that's like the specific feature or part of the entire virus that's oftentimes recognized as being the epitope against which the vaccines have been developed. Another set of terms to help define, we're going to talk about antibodies today. Antibodies are a protein that typically binds and recognizes epitopes. They'll recognize small amino acid sequences that serve as the epitope or a part of the antigen that they're meant to recognize. We're going to talk about a few different antibodies today, but it's first important to note what an antibody is, and that it's the key humoral component for actually adaptive immunology that will be, and that's the reason why we focus on it. Another key term that we alluded to last class, anaphylaxis. It's oftentimes tied to allergy. But it's this systemic reaction that it oftentimes leads to drop in body temperature, drop in blood pressure, can lead to shock. Shock oftentimes can be reduced down to insufficient blood flow through organs, where there's can be like organ function loss. And so anaphylaxis is oftentimes specifically tied to a large allergic reaction in response to some sort of allergen that leads to huge gross changes. For example, drop in body temp, drop in blood pressure. So when we talk about proliferation or cells that proliferate, what that really means is that cells are dividing and becoming more numerous, they're growing. And so that's when one cell becomes 2, becomes 4, becomes 8. And finally, differentiate or differentiation is when a cell becomes more specialized, both in its structure and function. And so last class, I showed this large lineage of cells that descends from the hematopoietic stem cell down to the common myeloid progenitor, the common lymphoid progenitor. Going down that lineage tree is a process of differentiation, going from one cell to the next, where it's more specialized. And we'll be talking about a few examples today about differentiation. Before we move ahead, I want to stop and pause. I want to stop and pause for a couple questions here. Yes. Yeah. So great question. So antigen is any substance that elicits an immune response. An autoantigen, whenever you see auto, just immediately think that means self. That's something I probably should have defined. So whenever you see auto, that typically means self. So autoantigen is some substance that originates from you, not from some pathogen, but some substance that originates from you that is an antigen that elicits an immune response, typically an autoimmune disease, where the self is recognized as foreign. So in autoimmune disease, there are going to be specific autoantigens that are critical to the pathogenesis of that disease. Yes. Yeah. So Guillain-Barré syndrome. Schwann cells are gonna possess an autoantigen in that disease. In type 1 diabetes, pancreatic beta cells. In arthritis, collagen. I'm trying to think of other good autoimmune disease examples here. In multiple sclerosis, what's the autoantigen in multiple sclerosis? This is evading me. Oh, in lupus, we'll talk about this as well. Your own DNA is actually the autoantigen in lupus. And so for lupus, DNA is everywhere. So a lot of the symptoms of lupus manifest everywhere. And so the autoantigen, again, an antigen is anything that elicits an immune response. We'll talk about different points in this lecture. In the next lecture, we'll talk about different antigens. For example, LPS, lipopolysaccharide, is an antigen. Lipoteichoic acid is an antigen. Myriamyl dipeptide is an antigen. It's a substance that elicits an immune response. The way they do that is they activate toll-like receptors. Yes. It's an excellent question. It varies by autoimmune disease. There are some autoimmune diseases that can actually be elicited by some sort of infection, in this case, Guillain-Barré syndrome. Interestingly enough, I talked about multiple sclerosis. I think this is accurate because for a long time we didn't quite know what the etiology was of that disease. But last year, there was 3 giant papers published that actually tied it to Epstein-Barr virus infection. That seems to indicate that that's an infection that also leads to autoimmune disease. And a lot of times, there's also genetic disorders, genetic variants that can make someone susceptible to autoimmune disease. So there's multiple factors at play. Why do infections cause autoimmune disease? There's multiple reasons why. One we're going to talk about today is this idea of mimicry, where the pathogen mimics something that's in our body. And when we raise antibodies against that invading pathogen, it also attacks what it resembles in our body. And so we're going to talk about that for Guillain-Barré syndrome. But it's a great question. And it can kind of vary from autoimmune disease to autoimmune disease. Yes. Yeah. So, boy, how much time do we have? What I mean by that is there's a process by which, and we're going to talk about this more next class, but I'm going to try and summarize this in like 15 seconds. So you can also like cut me off as well if I go too long. But basically, we have cells of our adaptive immune system that when our body is developing, when we're a growing fetus and when we're also developing our immune system at a young age, there might be some cells of our body that react to self. Those typically are eliminated during development because we don't want cells of our immune system that recognize self as foreign, that we don't want them to recognize collagen as bad, that we don't want them to recognize myelin as bad. They're typically eliminated during the development of our immune system. If there's some sort of issue with that process due to some sort of genetic disorder, that can promote the emergence of autoimmune disease by those cells sticking around. It'll make more sense when we talk about it next class, because I don't think we'll get to it this lecture, but it's a great question. I'm also noticing the time. We should march on. So this is we talked about this slide with respect to innate immunity last class. Each one of these points here in this lecture, the key take-home message for this right now is that this is orders of magnitude more true with adaptive immunity. Diversity. So with innate immunity, we are capable of recognizing specific patterns. We're able to recognize specific patterns, and there's a library of specific patterns our innate immune system is able to respond to. Our adaptive immune cells, by nature of the antibodies that they develop in our antigen receptors, can respond to trillions of possible epitopes. So the vast library of epitopes that our adaptive immune system can respond to dwarfs innate immunity in comparison. Specificity, again, by nature of how many possible trillions of combinations they can respond to, that means that they are incredibly specific down to the amino acid sequence level of what they can respond to. And finally, the memory, we talk about how innate immunity is genetically encoded and that memory is passed down. The antibodies and lymphocytes of our adaptive immune system develops a long-lasting memory that lasts on the order of years when they first encounter whatever that antigen is. So for example, when you were really young, at probably 2 or 3 years old, most people probably got a shot for the MMR shot, measles, mumps, and rubella. That's one that you typically don't have to get again, I believe, because there's long-lasting memory where the antibody levels, the antibody titers, that's another way of saying it, and the memory cells that stick around last for years. And so that's one way in which adaptive immunity is different than innate immunity. We talked about the humoral and cell-mediated component of innate immunity. Today, we're going to take some time to try and focus on the humoral and cell-mediated component of adaptive immunity. The humoral component, again, humoral means typically bodily fluid. Usually, primarily, we'll be talking about it with respect to blood. But it's also true for adaptive immunity as well that there are other bodily fluids that are a component here. The main portion is going to be antibodies. They're going to be antibodies that bind to and promote the inactivation of the pathogens that they recognize, specific epitopes on that pathogen. The cell-mediated response is going to be particularly cells. We usually refer to the cells of the adaptive immune system as lymphocytes. And so the lymphocytes of our adaptive immune response are the way in which they contribute to this adaptive immunity is that they can actively eliminate pathogen-infected host cells. So they'll recognize cells that have been infected and promote their death by a really elegant process called apoptosis. That's programmed cell death. So that's different than cells dying in response to some tissue injury. So if you break your leg or have a car accident, there's going to be gross cell and tissue death. What this is going to be cell by cell lymphocytes going in and interacting with cells. And if they find an infected cell, they're going to have a protein that they express that basically is like a tap on the shoulder saying, you need to die. And so that's a little morbid, forgive me. But It is a really elegant process of controlled, programmed cell death. So I mentioned a moment ago how we typically refer to the cells of the adaptive immune system as lymphocytes. As I mentioned last class, immunology is always shifting and changing, but we're going to try and focus on the big picture here. And so when, we're going to focus on the main key cell types of adaptive immune response. If you Google search, give me all the cells of the adaptive immune system, you get a lot more. And that's because as time goes on, we're identifying new novel cells. We're going to hone in on two large cell populations here, two types of lymphocytes. The first is B cells. B cells are the key cell responsible for our humoral immunity. That's because once they're activated and they differentiate, they specialize further into something called a plasma cell. That plasma cell is our professional antibody producing cell. They are the cells that secrete antibodies and help promote lasting memory. And so again, B cells will be naive. And then once they're activated, they'll differentiate into a plasma cell. And that plasma cell has the structure and capability of cranking out large amounts of antibody. They are a key antibody producing cell. And what's also important to note is that they are also one of our antigen presenting cells. And so on both sides of the board here, I listed out APCs, antigen presenting cells. So a couple of the cells listed here are ones that we talked about last class. There's shorthand that I've written here just because it gets kind of tiring to write some of these cells out. And this is kind of like the immunologist shorthand, so forgive me. But for APCs, antigen presenting cells, we have macrophages, dendritic cells, and B cells. We talked about phagocytes last class as well. The main phagocytic cells that we have are macrophages, dendritic cells, and neutrophils. So what's important to note here is that B cells are not phagocytes, but they are antigen presenting cells. And the way in which they serve as an antigen presenting cell is that they have this really large receptor that enables them to bind to and recognize very specific antigens. And they'll have receptor mediated endocytosis, where they'll then, and this is what all antigen presenting cells do at this point now, once they internalize, whether by phagocytosis for macrophages and dendritic cells, or just receptor mediated endocytosis for B cells, basically they'll chop it up into little pieces. You can imagine taking a large Lego structure and taking it down into its constitutive individual Lego pieces and display it. So these antigen presenting cells display chunks of the antigen that they've just engulfed or internalized, and that is antigen presentation. They are presenting antigen on the surface. If you're asking yourselves, what are they presenting to? Hold that thought because we'll get there. The key cell in cell-mediated immunity for the adaptive side are going to be T cells. So the two main cells that we have in adaptive immunity are B cells and T cells. If you're wondering why are they called a B cell and a T cell, we'll explain that later on today. But T cells are really key for direct cell-to-cell contact, and they're part of they're the ones that have a really key role in promoting that apoptosis, that programmed cell death. So they'll bind to and actually interact with infected cells or non-infected cells, trying to assess who is a healthy cell and who's not. And then they'll either deliver some sort of toxin, like something that's called perforin, that can actually poke holes and lyse the cell. Lysing, again, means like blowing up the cell, for lack of a better phrase, disrupting its osmotic pressure and destroying the cell, or inducing apoptosis, programmed cell death, where the cell will go ahead and die. And another key function for T cells here is that there's a flavor of them that help regulate and modulate the immune system. A lot of what we've talked about is me describing how different things can activate this part of the immune response, can activate this part of the immune response. If you're thinking about if we thought about the immune system as gas and brakes, everything we've talked about is all gas so far. And if the immune system was all gas, that'd be a huge problem. So we have a flavor of T cells that help serve as the brakes that help regulate things. And we'll talk about that flavor of T cells later on. One last thing that I want to touch on really quickly is that antigen presentation, when those pieces are chewed up and displayed, that process is really promiscuous, that they're going around and showing the antigen a ton. And again, we'll talk about what they're presenting to or what they're showing to. a key part of lymphocyte function is mediated by something called antigen receptors. As you can imagine, again, there's a lot of pieces here, but try and focus on the big picture and then populate in the details. Antigen receptors are really important because they are the receptors on our lymphocytes that bind to antigen. Both the humoral and cell mediated components of both B cells and T cells express and heavily rely on their antigen receptors for their function. As you can imagine, again, as the name implies, an antigen receptor, the way it works is it's a receptor whose ligand is an antigen. And this is what's really, really important for adaptive immunity here. This antigen receptor on the surface of a T cell or a B cell binds to one and only one antigen. Not only that, it binds to one and only one epitope. A single T cell or a single B cell might have 20,000 copies of that antigen receptor, but on that one B cell or on that one T cell, they have one and only one copy that they express. We're going to have multiple clones of that T cell or B cell with that antigen receptor. For example, again, of all the vaccines that we've all had over the years, we have lots of memory cells circulating that have multiple copies of that antigen receptor against the MMR vaccine, against Hep B, against the litany of vaccines we've all had. But each cell has one and only one copy littered across its surface. Again, about 20,000 copies, but one and only one. And antibodies are a class of antigen receptor. What's important to note here is that well, I won't dive into that quite yet. So antibodies are a class of antigen receptor, and the antigen receptors actually resemble an antibody in a lot of cases. So I described how there's one and only one copy on each antigen, on each lymphocyte with for their antigen receptor. So that means their entire destiny is to find that antigen or the epitope on that antigen that they're meant to recognize. That is their fulfillment in life, for lack of a better phrase. And so because throughout different regions of the world, there are different antigens that we're all exposed to, you might have copies of antigen, you might have antigen receptors that never interact with the ligand that they're meant to bind to. And the ligand, the antigen that they're meant to bind to, we refer to as their cognate antigen. So our antigen receptors have a particular cognate antigen that they're destined to interact with. And so there are a lot of cognate antigens that our body, that our cells, our lymphocytes have already seen. There are some that they have not seen yet, and there are some that they might not see just by nature of the environment we live in and what kind of antigens we're exposed to. I'm going to hit pause here and give space for a couple of questions. Yes. Great question. A single antigen can have many epitopes on them. And oftentimes, there might be different antibodies that recognize different epitopes that bind to the same antigen. Meaning, for example, if I was a pathogen and I invaded someone's body, for lack of a better phrase, there might be an antibody against my wrist. There might be an antibody against my elbow. There might be an antibody against my leg. So like there's different antibodies can recognize different parts or different epitopes of the same antigen. That was a poorly worded metaphor as I thought about this now. So please forgive me as this is a recorded lecture. Yes. Yes.
Even if like the disease is not.
To make sure I understand your question, some things of our immune system, some things of the immune system is inherited, and some things we are, for lack of a better phrase, given to through breast milk for young children.
If like for 4 generations, for example, like an individual is not a certain antigen after, is that how long is it still
For that antigen receptor? Yes. Okay. Thank you. Great question. By nature of the genes, that are responsible for generating these antigen receptors and how they change during some key processes. We're gonna talk about the process. It's also a wildly complicated process, but it's basically a library of genes where there are changes that happen where we develop a large catalog of possible antigen receptors that you have at birth. So it's not the antigen receptors that you have typically are not something that's inherited, but rather a encoded feature of the genes that are responsible for antigen receptors. Does that help answer the question? That was a great question. I appreciate you asking that. I'm going to keep marching forward just to make sure we at least get to the stopping point I hope for. We might not get there, but we'll press on.
One big thing
Yeah.
is that the immunity that's passed from like mother to baby is in the form largely of antibodies. And it's designed to basically cover the period of time that the baby's not making its own antibodies. And so those are likely to be antibodies that the mother has like generated either because of exposure to certain things, because of vaccinations to certain things. There are even some vaccinations that are designed for women who are carrying the babies so that they can generate those antibodies and pass them on. Just so that we don't confuse the antibody transfer from mother to baby with like any kind of genetic encoding of immunity, which is what also that recombination process.
Perfect. Thank you. I appreciate that. So here we have the structure of an antibody. So an antibody is a tetrapeptide. Tetra means four. So it's a four-part peptide where we have two light chains and two heavy chains. And so I've also tried to depict on the board on both sides where we have our light chains and heavy chains. And so we have basically a simple depiction of an antibody typically is a Y-shaped molecule. A more biologically and chemically, a more biochemically accurate depiction of an antibody is there are, try not to get too far away from the microphone. We have a light chain on the outside here and a light chain on the outside here with two heavy chains on the inside. So basically you can think of a Y-shaped molecule with two short arms underneath that are the light chains. So we have two light chains and two heavy chains that make up the tetrapeptide. Both light chains and heavy chains have something that's called a variable and a constant region. As you can imagine, because we talk about so many different possible copies of antibodies, because it's an antigen receptor that can possibly be made, it's the variable region that changes that helps provide that diversity, and the constant region remains constant. And there's a reason why, and we'll talk about it shortly. What's also important to note is that there are the different regions of an antibody have different functions, and that's, there we go, depicted here as well as on the board. So we have our FAB region. What that stands for is antigen binding region, and we have our FC domain. The C typically means, I believe it's crystal domain. What helps me in terms of helping understand its function, kind of like with AB means antigen binding, the C region actually binds to FC receptors on cells. So there are receptors on cells they have for antibodies, and it's the FC portion, C meaning cell, that binds to these receptors on cells. And so as you can imagine, as you can probably, just to try and head off any questions, that means within the FAB region, within the antigen binding or FAB region, we have part of the light chain and the heavy chain within it. And there's also going to be part of the variable and the constant domain within it. And so we're going to talk about a little bit further about why that's beneficial when we talk about the antigen receptors and their diversity in a little bit. Looking at a depiction of ribbon depiction of the antigen binding domain, the FAB domain, what we have here is that there's a specific region called the antigen binding site or the antigen binding pocket, which is like the Y arms on the antibody, where the antigen will actually be bound to at the epitope that this antibody recognizes. And again, we have part of the light chain and part of the heavy chain within this antigen binding region that helps contribute to make up what recognizes the epitope on the antigen that they bind to. I'm going to stop here and give space for a couple of questions. Yes.
Where would this be like on a bigger scale?
Yeah, so this is just a depiction, this is a ribbon depiction of the FAB region. So the antigen binding pocket literally is out here at the top of the antigen binding site where the pocket is, where the binding site is. You can imagine some of the most endearing immunologists I've met will sometimes have dreams that they're antibodies, and you can imagine like the antigen binding domain is like your arms. It's super nerdy, but they're super endearing. But you can imagine the antigen binding region is like your hands if you were an antibody yourself. That's the antigen binding pocket. There's a reason why I said that, because it kind of helped with the hands. Sorry, go ahead. Great question. This is one antibody, and every antibody has two antigen binding sites. They're identical, but they're two binding sites. And what that enables is if there's an opportunity to bind to multiple antigens where the same epitope is available, like that's kind of one of the strengths of having the two arms of the antibody. So this is one antibody with both antigen binding sites, the FAB regions depicted. They bind to they both recognize the same epitope. They're both identical to one another. I'm gonna go here and here. Go ahead. So great question. So the heavy chain is part of both the FC and the FAB region. And that's kind of all, that's what I was trying to depict in my illustration here, though it might not be fully appreciated. You have, this is the heavy chain. So this is part of the FC domain and part of the antigen binding domain, the FAB domain. Did you, go ahead. The light and heavy chains are, no, they're both identical. Yeah. All right, we're going to press on. So we have our, again, our antigen binding pocket that's depicted here in the FAB domain. This goes back to a question earlier about antigens having multiple antibodies bind to them. When we have multiple antibodies, well, I'll wait there. Antibodies are multivalent, meaning they can bind to multiple antigens. They have two FAB domains, meaning they can bind to one antigen here and one antigen here. What ends up happening is when we have both arms, when we have two antibodies binding to an antigen, that's called antibody cross-linking. And so when we have multiple antibodies binding to a single antigen, that's typically referred to as an immune complex. Again, when you have multiple antibodies binding a single antigen, we'll typically refer to that as an immune complex. This can be really helpful in a research setting because, yeah, you want to go ahead and ask a question? Same a proteoglycan would be.
Yeah.
The process of just cross-linking is when two antibodies bind, or when two antibodies are binding to one single antigen. So it's not entirely similar to what works with proteoglycans. There's going to be a few different examples we're going to provide of antibody cross-linking that are kind of key in biology, but hold that thought. And I'll get to it in a second. Hold on, let me finish the slide. I appreciate all the questions. The way we leverage immune complexes in a research setting is if we want to purify for a specific protein, we'll use we'll use antibodies that will again form an immune complex around whatever the antibody recognizes, whatever the antigen is, and it will centrifuges down to pull down and precipitate out whatever the immune complex is recognizing. What's really key, and we have it depicted here, where there's cross-linking of multiple antibodies on a single antigen and the formation of an immune complex around these antigens. What's really important about these immune complexes, that's key, is they can help promote activation of the complement system. We talked about last class how when antibodies bind to a pathogen, it can recruit C1q that will drive the classical pathway of complement. It'll actually drive phagocytosis. So I mentioned earlier how the FC domain of an antibody, that some cells have a receptor for FC. We call them FC receptors. And when an antibody is docked on that FC receptor, and if it's bound to a specific antigen, that can actually promote in particularly macrophages and neutrophils, but we're also learning about more cells that can participate in this. They'll promote the phagocytosis of whatever the immune complex is formed around. And finally, it can actually promote a unique thing called degranulation. So this kind of goes back to your cross-linking question. There are certain cells that are called granulocytes. Three key cells are mast cells, neutrophils, and eosinophils. I did my PhD in a mast cell biology lab, so I think it's the coolest granulocyte, but most immunologists beg to differ. What's really unique is that mast cells are one of the key cell types that are involved with allergy. So they're unique in that they have an FC receptor for a certain class of antibodies. And usually the way you have an allergic response to something is that that antibody recognizes something that's innocuous as actually dangerous. So for me, that's cat dander. So whenever my mast cells encounter cat dander, these granules, these little pockets that have lots of biomolecules in them, degranulation is the sudden release of the granule contents, which is usually histamine, lipids, and inflammatory proteins. And so the degranulation is triggered by these antibodies binding to an antigen, cross-linking together on this allergen, and promoting the rapid degranulation release of the granules that usually consists of histamine, lipids, and inflammatory signaling proteins. All right, I think we're going to get to where I was hoping to, and I'll have space for a couple questions afterwards. I'll scoot myself a little bit here, but the activation of cells that are not lymphocytes via these antibodies oftentimes occurs through FC receptors. Again, I use the example of mast cells. They have, using this depiction here, on the surface of a cell, there's a receptor that's called an FC receptor. It binds to the FC domain of an antibody. And so in some cases, they'll already be bound to whatever the antigen is they recognize. In the case of mast cells, all of our mast cells have IgE already, an antibody already on the surface, waiting to interact with their cognate antigen. For some of you, it might be some sort of food allergen. In some cases, it might be pollen. And when that happens, the cross-linking of these antibodies on the surface on this allergen triggers, for these granulocytes, massive release of their granules fairly quickly. And in some cases, phagocytosis, if this antibody is a certain flavor of antibodies. When I talk about flavor of antibodies, we actually have 5 flavor of antibodies. The most abundant is called IgG. So the IG stands for immunoglobulin. That's a longhand term for antibody. So if you ever see immunoglobulin, that typically means that there's a certain structure that resembles antibodies here. So in this case, whenever we talk about IgG or any of the antibodies, it'll be the most abundant one is IgG. And the FC receptor for each of them, it's not a wild nomenclature, but it's pretty easy to follow along. It's usually FC and the Greek letter of whatever this letter is here. So for IgG, it's called the FC gamma receptor. The next most abundant one is IgD. That's typically an early version of the antigen receptor on our B cells called the B cell receptor. So the T cells have a T cell receptor, B cells have a B cell receptor. I've depicted the B cell receptor on both sides here. It's literally an antibody wedged into the cell membrane. That's typically what the B cell receptor resembles. And so an early version of the B cell receptor is actually IgD. The next is IgM. That's typically the flavor of antigen receptor you'll find on B cells. And I just realized I'm looking at my screen, not looking there. IgM is typically the flavor of antibody you'll find on the surface of B cell. And it's the first type of antibody that will be generated. When it matures in response to like a secondary exposure, it'll mature and class switch. We'll talk about class switching later. So just kind of keep that in the back of your mind. Class switch to IgG, which is why most of the antibodies that you'll find in your blood are usually IgG. Next is called IgA. IgA is typically what you'll find in your mucosal layers. So for example, I think I talked about in a previous lecture about new generations of the COVID vaccine that are meant to be more appropriate for the context where the virus will be infecting. Most of our antibodies that are raised against the current COVID vaccines are usually IgG. But the mucosal layer antibodies are usually IgA that are now the next generation of COVID vaccines are meant to try and raise against that. And the last one, and certainly not least, is IgE that binds to mast cells and basophils, but trigger degranulation that are associated with the classic allergic reaction. The order of abundancy, or the order of abundance here, is GMADE, GMADE. That's the order in which you'd expect to see if you order them in terms of how abundant they are. That's the order in which they occur. And again, the Fc receptors is Fc and then the Greek letter of the blue letter. So that means we have Fc gamma, Fc delta, Fc mu, Fc alpha, and Fc epsilon for each. There are the receptors for each of these antibodies here. I think I'm going to go ahead. Oh, and I should one last thing. This is the slide I wanted to end on. These different antibodies can form different immune complexes. So IgG is typically a standalone monomer. IgM with a linking protein can actually form a pentameric structure and I didn't depict here, IgA actually forms a dimer as well. And so this is just to depict how the different ways in which these antibodies can emerge in biology. I'm going to stop the recording there, but I'm also going to give space for questions as well if people have questions. You're also allowed to just go ahead and roll out too.